Welcome dears, welcome to the program Toppers NEET and Olympiad Foundation course, and the class is 9th and our topic is a continuation of reproduction in flowering plants. So today we are going to discuss some of the previous content questions. Okay, so let us see the continuation question of 66. So today we are going to start the question from the 67th question. Let us see what is the question here. The pollen grain is what is the pollen grain condition? Okay, so option A, what he gave, an immature male gametophyte. Option B is a mature male gametophyte. Option C is a partially developed male gametophyte, and option D is a last stage of male gametophyte. Okay, so let us see what is the correct option here. Before going to the options, so if we observe the picture over here, so this is the pollen tube, sperm cell and the vegetative nucleus, pollen tube. Okay, so within this is the pistil region. Okay, so within the pistil region we can observe the germination of a pollen tube. Okay, so the pollen tube contains two sperm nuclei, and at the bottom we can observe one of the nucleus, and that will be developed by the vegetative cell which develops the pollen tube. Okay, so here we can observe the sperm cell will be penetrating, is nothing but the germination of pollen tube will be done in the pistil region. Okay, so before to that we can observe, so how does it forms, how this germination process forms. Before the germination process, the pollen grain has to develop. Okay, after the pollen grain it has to burst open. Okay, so before the bursting condition it is in a immature condition. After the bursting it is considered as a mature gametophyte. Okay, so this is the microsporophyte which divides meiotically and forms tetrad. Nothing but four pollen microspores will be forming. So these microspore will be releasing the microspores. Okay, so after finishing the microspore formation we can observe the division. Previously it was in the meiotic cell division and now it is in a mitotic cell division. We can observe here the vegetative nucleus and the generative nucleus. Generative nucleus divides mitotically and forms two sperm cells, and the vegetative nucleus will be responsible for the germination of formation of a pollen tube, and that germinated tube will be releasing towards to the pistil region in the female organ. Okay, nothing but the pistil. Okay, so after this the pollen will be dividing in a second process, mitosis. Mitosis 2. Mitosis 1 will be developing the vegetative and the generative cell. Mitosis 2 will be forming the two mature pollen cells. Okay, so here the condition is, so we can observe after the division of a mitotic cell division, the pollen grain has to burst open. Okay, so until it is in a grain, it is in a sac condition, the pollen grain is in a immature condition. Okay, so option A will be the correct one. The pollen grain is in a immature male gametophyte and it is a mature male gametophyte. When it is in a sac, it is not in a condition of a maturity level, it's not a correct option. And coming to option C, a partially developed male gametophyte. Okay, partially in the sense half. Okay, so option A and option C might be the correct ones. Okay, so in this we can observe that we can opt option A and option C. Coming to the last one, it's a last stage of male gametophyte it seems. No, after forming the pollen grain it has to burst open, so then we can observe the pollen tube will be generating by the germ cell, a vegetative cell. This is not the last stage. Hope you all understood dears. Coming to the next question. In flowering plants, a mature male gametophyte is derived from a pollen mother cell by. In a flowering plants, a mature male gametophyte is derived from a pollen mother by which kind of cell division he is asking. Okay, so if you observe this, so we can observe how many meiosis and how many mitosis we can observe here. Okay, so in the previous question also we have discussed, first meiosis division occurs for the formation of immature male gamete or the female gamete, and next we can observe for the division of sperm cells in the male, we can observe the mitotic cell division. Okay, so first meiosis and next mitosis. Okay, let us see here what are the correct options out of these. So option A is 3 mitotic divisions, option B is 1 meiotic and 2 mitotic divisions, option C is 2 meiotic divisions, and option D is a single meiotic division. Okay, so which one of these will be the correct option? As per the sequence over here, the microspore mother cell will be dividing meiotically first of all only for the one time. Here he has given meiosis 1 and meiosis 2. Those are the stages, but the division will be only single. 1 meiosis. And next to that we can observe the formation of a tetrad condition. So here the pollen grain in a tetrad condition, they will be separated by the mitotic cell division. So after formation of a mature pollen grains, we can observe that the pollen grains will be bursting open and then releasing towards to the stigma for a fertilization process. So which one will be the correct option? Option B will be the correct. Okay, not 3 mitotic divisions, not 2 meiotic divisions, and not a single meiotic division. Okay, hope you all understood, dears. Option B will be the correct one. And coming to the next question, when the ovule is curved and embryo sac becomes horseshoe shaped, such an ovule is called. Okay, so within the type of ovule, here the horseshoe shaped embryo sac is formed in which out of these type of ovules he is asking. Okay, so this is the orthotropous ovule. Horseshoe shape in the sense will be like this. Okay, so this kind of shaped embryo sac can be seen in which ovule? Okay, so is that in the orthotropous? It's completely wrong. Is that in the anatropous? No. Is that in the hemianatropous? No. Is that in the campylotropous? No. Exactly seen like this. Okay, so here we can observe the amphitropous ovule. The embryo sac shape is resembling exactly like a horseshoe shaped structure. Is that a circinotropous? Exactly wrong. Okay, so out of these options, which one will be the correct one? The amphitropous ovule, not the circinotropous, campylotropous and orthotropous, as we have seen in the image. So this amphitropous will be the correct option, but not the remaining ones. Okay, next. Coming to the next question, what is embryo sac in this condition? Okay, embryo sac is a megasporangium. It's a megaspore or a female gametophyte or a female gamete. Okay, the picture indicates it's a female gamete. A mature embryo sac is a female gamete. Okay, it's not a megasporangium, it's not a megaspore. What is megasporangium? Sporangium can be seen in the spore structures. Okay, megaspore mother cell, that is during the developmental condition of a female gametophyte. Okay, so option C will be the correct one. The embryo sac is a female gametophyte. Okay, but not the female gamete. What is the difference between female gamete and the female gametophyte? Female gamete is the ovule, female gametophyte is the embryo sac. Matured embryo sac is the female gametophyte. A female gamete is the ovule. Okay, next. Coming to the next question. What is the next question? In anatropous ovule, the micropyle is. Where the micropyle in the anatropous ovule he is asking. Okay. So before going to the options, let us see the position of the micropyle in anatropous. Out of all these, where is the anatropous ovule? B1 is the anatropous ovule. Okay. Side towards to the funicle, not the straight line condition. Okay. So in a anatropous ovule, the micropyle is side by side with the funicle. Okay. So here the funicle is straight, both are straight, but not funicle, but not the micropyle is in this condition. It is in a side by side condition. Okay. Not the straight line condition, right angle of the funicle, exactly at the 45 degrees of the funicle, but it is in a side by side towards to the funicle. Okay. Understood? Next, coming to the next question. 72 might be the repeated one. Let us see, the ovule is in a P shape R. Okay, so what are the P shaped? This is the P shape. Okay, so P shape is attached towards to the, like this. Okay, so this kind of ovule is seen in which out of these ovules he is asking. The ovule is P in PR, anatropous, hemianatropous, campylotropous or the amphitropous. Okay, so see in the picture, what is the option A? It's a anatropous. Okay, so where is the anatropous? Option B is the anatropous. Is this shape similar to this shape? Exactly no. And coming to the option B, hemianatropous. See, this is the hemianatropous. Okay, so is that the same shape of a P shape? Not exactly, right? And coming to option D1, you see campylotropous, exactly coming in a same shape. Okay, same shape, a spear shaped structure. Okay, so this is looking like a P shape. So what is the D1? It's a campylotropous ovule. Coming to the options, option C is the campylotropous ovule, which is exactly in a P shape, right? And coming to amphitropous ovule is also not the correct one. Coming to the next question, in an embryo sac of a typical angiosperm there are, so what are the type of cells? Count of the cells he is asking here. So type of cells, not the count. In an embryo sac of a typical angiosperm there are eggs, synergids and the antipodals. So before going to the options, let us see the picture over here. First of all, here we can observe at the posterior part the two synergids on either side of the egg cell, and in between we can observe the egg cell. Okay, so two synergids and one egg cell, and in the middle region we can observe the central cell with two polar nuclei. Coming to the anterior region, we can observe three antipodals. Okay, three antipodals, one central cell with polar nuclei and one egg cell. Either side of the egg cell we can observe two synergids. Okay, so where we can observe those structures? Egg, synergids and antipodals. Here something is missing. Central cell is missing. Egg, yes. Synergids, yes. Polar nuclei, also called as a central cell, and the antipodals. All the four are here, involved here. And coming to option C, egg, synergids, central cell and polar nuclei, but antipodals are missing here. Yes, and coming to option D, egg, synergids and secondary cell. Okay, so again the antipodals are missing. Okay, so what will be the correct options? So by this we can conclude that option B will be the correct one, not option A, not C and not D, right? Coming to the next questions here. A mature polygonum type of embryo sac has got, right? A mature embryo sac, polygonum embryo sac has got how many cells and how many nuclei? It's a very easy, simple question, okay? So let us see what are those. 7 cells and 8 nuclei. Exactly. 3 plus 1 plus 3. 3 plus 3 is 6. 6 plus 1 is 7. Okay, but coming to nuclei, let us see one count. 1, 2, 3, 4 and 5, 6, 7 and 8. How many nuclei? Nuclei will be 8 in count. Yes. And cells will be 7. Okay. So, by the first option only we got to know that yes, option A is the correct one. Coming to the next options, let us conclude these also. 7 nuclei and 8 cells. Completely opposite to this he gave. Okay. It's a wrong option. Coming to option C, 8 cells and 8 nuclei. No similar cells and nuclei will be formed. Nuclei are one extra than the cells, right? And coming to option D, 7 cells and 7 nuclei. No, completely wrong. So, which one will be the correct option? 7 cells and 8 nuclei, right? Next, coming to the next question. Egg apparatus of angiosperm consists of egg apparatus. What is egg apparatus? Okay, so combination of 3 cells. One is the egg cell and 2 more are the synergid cells. Okay, so let us see here, 1 egg cell and 2 synergids. Yes, obviously correct. 1 egg cell and 2 synergids and 3 antipodal systems. Antipodals where we can observe in the anterior region. Okay, so in the egg apparatus, the combination of only synergids. How many synergids? Only 2 synergids will be seen and only single egg cell can be seen. Okay, so if we observe this, so this will be considered as a egg apparatus. The picture indicates synergids, in the sense the filiform cells and the egg apparatus. Okay, so here we can observe the filiform apparatus and the synergids. It must include the egg cell also. Okay, so let us see here, 1 egg cell and 2 synergids and 3 antipodals. No, 3 antipodals only. No, secondary nucleus and the egg cell itself. Secondary nucleus will be in the central region, and coming to secondary nucleus, it's completely wrong and only egg cell itself. Wrong option. So, this must include the egg cell, synergids, and within the synergids we can observe the filiform apparatus. Okay, so that includes the egg apparatus. Option A will be the correct one. Option B, C, D are the wrong ones. Okay, next, coming to the next question. An ovule which becomes curved so that the nucellus and embryo. The ovule is curvy structure, it seems. So, by the curvy structure of the ovule, so we can observe the nucellus and embryo sac lie at the right angle towards to the funicle. Okay, so where will be the funicle here? To the funicle, the embryo sac and nucellus are exactly right angles, it seems. Okay, so what are those? This is the funicle, right? And this is the embryo sac. The entry point is the micropyle. And where we can observe the nucellus? Surrounding the embryo sac we can observe the nucellus. Nucellus and the micropyle are exactly in a straight line in the first ovule. Okay, so this is a wrong option. And here also the nucellus and micropyle are straight line, exactly side by side conditions towards to the funicle. This is also wrong. So, this doesn't make the right angle. And coming to the amphitropus, we can observe the funicle and micropyle and nucleus. Not exactly, it's just a slight condition, slight sliding condition it is. So, this is also the wrong option. Anatropus and atropus ovule is also wrong. hemianatropous. Okay, this is the funicle. And the micropyle and nucellus. Micropyle and nucellus are exactly perpendicular in a right angle direction. Okay, so hemianatropous ovule will be the correct option, not the camphilotropous. No. Cercinotropous, exactly this is opposite. Funicle, micropyle and nucellus. This is left hand side. Okay, side by side but left hand side. Here also side by side but right hand side. Funicle is right side and nucellus with the micropyle is at the left hand side. Coming to cercinotropous ovule, we can observe the funicle is in a left hand side. Right hand side we can observe the inverted region, we can observe the micropyle and the nucellus. Okay, so out of these, which will be the correct option? The hemitropous ovule will be the hemiana or hemitropous also we can say. This option C will be the correct one, not the anatropous, nor the orthotropous and not the camphilotropous. Okay, right. Coming to the next question, a stalk with which ovules remain attached to the placenta. A stalk with which, okay, so what is a stalk? A part which can be holded. Okay, so holding two regions. Okay, so what is that stalk with which ovules remain attached? Ovule is remaining attached to the placenta. This is the placenta. Okay, the ovule is attached towards to the placenta. What is that condition called as? Is that the funicle, a micropyle or the hilum or the chalaza? Okay, so which one is the correct option? Let us see here. This is the funicle. The stalk is attached. The holding part is called as a funicle, is attaching towards to the placental region. Okay, so this is the correct option. What is attached towards to the placenta? A stalk like structure called as a funicle. Okay, option A will be the correct one. Okay, so why not the micropyle? Micropyle is exactly side condition. Okay, side towards to the funicle and there is no joining of placenta and the micropyle, right? It has to be attached. The word itself indicates, okay, attaching towards to the placenta is the stalky structure called as a funicle, not the hilum. Where is the hilum? Hilum we can observe the structure which is connecting link towards to the chalazal and the funicle. Okay, so this is called as a hilum. The connecting link between the funicle and the complete ovule is called as a hilum. Okay, this is also the wrong option. Micropyle also will be the wrong option and chalaza. This is the chalazal end. Where is the placenta and where is the chalaza? Completely wrong option. Okay, so which one will be the correct option? Funicle is attached towards to the placenta and this is a stalk-like structure. What structure it is? It is a stalk-like structure which must be attached towards to the placenta. Understood? Right. Coming to the next question, the point of attachment of a funicle with the chalazal end is called a point where it is attaching both the funicle with the chalazal end is called as placenta. Not exactly. Placenta will be at the basal region. The stalk-like structure is called as a funicle region. Okay. Integument, not completely. Integument can be seen in this region. Okay. Inside to this we can observe the embryo sac. Okay. So is that the nucellus? Inside the embryo sac, outside the embryo sac we can observe the nucellus. Okay, so see, this is the nucellus. Surrounding the embryo sac is the nucellus and what is the region? Placenta will be at the basal region of the stalky structure called as a funicle. And what is the hilum? Hilum is attaching both, the point where it is attaching both the funicle and the chalazal end. Where is the chalazal end? This is the chalazal end. Okay, so here the chalazal end is continuing from the funicle. Okay, so what is the adjoining point is called as a hilum. Okay, what is it called as? It's a hilum, not the placenta, not the integument, and not the nucellus. Hope you all understood, dears. Right, so according to the picture, if you got to know, so then easily you can observe. So immediately you need to think. In exam you will not have a picture. Here just for the conclusion, just for the clear explanations, we are just giving the correct option as a pictures here. But in exam what you need to do? Just you need to think of what are the parts which are present in the ovule. Okay, so this is the placenta, this is the funicle, this is the chalazal end, and inside gametophytic cell is called as a embryo sac, and these are surrounding the nucellus are the integumentary regions, and like that we need to just think of it in our mind and immediately we need to go for the option. In exam will not have a correct time, proper time will not be given there. Okay, next. The term used for transfer of pollen grains from anther of one plant to stigma of a different plant. Okay, so which during the pollination brings the genetically different types of pollen grains. Okay, so nothing but pollination, we know that there are two types of pollinations. One is the self-pollination within same flower, or within the same plant different flowers will be seen. That is called as a self-pollination. And what is cross-pollination? The transferring of pollen grains from anther to the stigma of the another plant or another flower is called as a cross-pollination. But here we are observing pollination, transfer of pollen grains from the anthers of one plant itself, one type of plant. Okay, type of plant to the stigma of a different type of plant which during pollination, so that brings, so if this process occurs in a different types of plants, so that is bringing a kind of change it seems. Okay, new genetically modification types of pollen grains are being formed. Okay, so this process is called, is that the chasmogamy or a cleistogamy or a xenogamy or a gitmogamy? Okay, so which one will be the correct option? Option C will be the correct option, xenogamy. So in this we can observe a genetically modified in the sense generally occurs. Okay, not by the man-made. So here we can observe the different pollen grains genetically different. Okay, so these are nothing but the process is called as a xenogamy. And what is chasmogamy? Chasmogamy in the sense, so here we can observe the exposure of the pollen grain towards to the stigma. So here we can observe either self-pollination or cross-pollination. There might be a chances of both self and the cross-pollination in the chasmogamy. And coming to cleistogamy, what is cleistogamy? So within the closed flower we can observe the process of a pollination process. Unopened type of flowers we can observe the cleistogamous pollination process. And what is xenogamy? This is the definition part. Okay, and what is gitmogamy? Gitmogamy is a type of a self-pollination process. So here we can observe flowers will be different but the plant will be same. Okay, so this is called as a gitmogamy. Gitmogamy is not, no, they don't produce the genetically different types of pollen grains. Only in the xenogamy process we can observe the genetically different types of pollen grains. Right? Hope you all understood, dears. Thank you so much. Hope you all enjoyed this.